Hello students, and welcome to our final lecture in our microbiology series. This is lecture 12. And in the previous lecture we described methods of killing bacteria on surfaces and other places outside the body. So we learned various methods that we could use to kill bacteria, for the most part bacteria, but other microbes on the surface of the skin and so on, and that usually involves chemical methods like povidone iodine tincture. Also killing microbes on surfaces, food preparation surfaces and so on, and everything up to and including sterilization in an autoclave, which is virtually guaranteed to kill everything. But obviously, if the bacteria are inside the human body, then we have to resort to mechanisms that can kill the bacteria without harming the human. And so these are, if we're talking about bacteria, we call them antibiotics. Antibiotic drugs are drugs that kill specifically bacteria. The reason for that, the reason I specify bacteria, is because if you're going to kill something that's invading the human body, without killing the human, you have to exploit something that's different between the human and the microbial invader. As it happens, luckily there are a lot of differences between the human body and the bacterial body, but there are very few differences. There are fewer differences between humans and protists, and there are fewer differences between humans and yeast and fungi. You heard me mention early on in the course that fungi, humans reinforce their plasma cell membrane with cholesterol to make it more rigid. Fungi use a related chemical called ergosterol to reinforce their plasma membrane and make it more rigid. And so antifungal agents, those are chemicals that kill fungus. Antifungal agents will essentially block the enzymes that build ergosterol while at the same time they leave the enzymes that build cholesterol alone. So therefore we've specifically poisoned fungus with various chemicals that are able to poison or inhibit the enzymes that the fungi use to build ergosterol. Okay, well, that's fungus, but as it happens, there are a lot of differences between bacteria and humans. The most conspicuous one is the peptidoglycan cell wall. So there are a lot of drugs that destroy or inhibit the formation of the peptidoglycan cell wall, and therefore they kill bacteria, specifically gram-positive bacteria. Also, bacteria have different ribosomes than humans. Remember, humans have an 80S ribosome, while bacteria has a 70S ribosome. So there are many antibiotics that are chemicals that bind to and deactivate the bacterial ribosomal subunits. They bind to and inactivate the 30S or the 50S subunit of the bacterial 70S ribosome. Those are the main ones. There are a few others, and we'll go through those as we go. So let's talk about some of the things that kill microbes on the inside of the human body. All right, so this lecture used to be divided into 4 parts, talking about things that kill bacteria, things that kill fungi, things that kill viruses, and things that kill protozoa and helminths. I think we've done enough for this semester, so we're really only going to deal with things that kill bacteria. If you're interested, there are some in the class notes, in the notes for the course, there are some notes about various chemicals that kill fungi, protozoa, helminths, and I'm hesitant to say kill viruses because obviously viruses are not alive in the first place, but there are some specific chemicals that are called antimicrobials, or sorry, antiviral chemicals, that inhibit viral reproduction is what they do. And there are a few drugs that do that. In my opinion, they're not nearly as good as having a vaccine. If you have a vaccine, that prevents you from catching a viral infection in the first place, and that's far better than catching a viral infection and then having to rely on some chemicals to sort of do a lousy job of trying to inhibit the viral replication cycle. All right, so let's just concentrate on bacteria. All right, so specifically, drugs that kill bacteria are called antibiotics. Antibiotics. All right, so ideally there are 3 ways to deal with a bacterial infection. Number 1 is you can take probiotics. As you know, the bad microbes like Clostridium difficile usually are excluded from the human intestine, for example, because there are too many good bacteria in the human intestine that will not give them any room to set up house. So bad bacteria are excluded from the human intestine by competitive exclusion, meaning that there are so many good bacteria there already that there's no room for the bad bacteria like C. difficile to move in. So one of the ways that you can avoid getting bad infections in the digestive system, for instance, is to take probiotics. So probiotics are a little bit controversial at the moment. Some people say that they don't do as much as they claim to do. There are a lot of pharmaceutical companies that are selling pills that contain lyophilized E. coli and a mixture of lyophilized E. coli, Streptomyces cerevisiae, and Lactobacillus acidophilus. Those are usually the big three. There's some other ones that they put in there. And you take these pills, these lyophilized bacteria, and they come out in the intestine and they get rehydrated and they come back to life. And theoretically and ideally, they should exclude the bad bugs from being able to move in. All right, that's one way to deal with avoiding getting a bacterial infection in the first place. Secondly, you're probably already aware that vaccination is not just for the prevention of viral infections. There are many vaccines that will prevent you from catching bacterial infections as well. So there are many vaccines that prepare your immune system to kill certain bacteria when they enter your body. Okay, but if you haven't done either of these things and you're unlucky enough to get invaded by bacteria anyway, we have the antibiotics. So these are drugs that selectively kill the bacteria while leaving the human host alone. In some cases, there are some bacteria that do a very good job of killing. There's some antibiotics that do a very good job of killing the bacteria and do absolutely no harm to the human. They're not toxic at all to the human. But as you know, some bacteria will develop a resistance to these antibiotics, and then you have to move on to another antibiotic that will kill the bacteria but will also do some damage to the human host as well. Right, so that's the worst option. If we have bacteria that are developing resistance to the easy antibiotics, we have to move on to the more difficult antibiotics, which is unfortunate. And there are some antibiotics that are so toxic to humans that yes, they will kill bacteria in your system, but they will damage your liver. They will do great damage to your liver. In some cases, they'll damage your hearing and so on. And so we don't even use those to kill bacteria in humans. We sometimes use them to kill bacteria in animals because as a group, humans care less about animals than they do about other humans, most of us. And then in some cases, there's some antibiotics that are so toxic that we only use them in the laboratory. Basically, there are some antibiotics that are used in the laboratory to keep bacterial cells from contaminating tissue cultures. So when we grow human cells or animal cells in a bottle, in a flask, that's called tissue culturing. You heard early on in the course about HeLa cells, for instance, which came from a woman named Henrietta Lacks. When we do tissue culturing, remember that human cells will, on average, in a tissue culture, human cells will divide every 24 hours, once a day, whereas bacterial cells will divide every 20 minutes under those conditions. And so if you have a human cell culture that's contaminated with bacteria, that's really bad because the bacteria will be dividing every 20 minutes and they will just take over in a flash, and the human cells will be killed off by all these bacteria. So in the laboratory, in tissue culturing, we add antibiotics to tissue culture flasks to keep bacteria from growing in, with the human cells and other animal cells. But those antibiotics are quite toxic. But we don't care because we're not putting them inside a human being. We're only putting them inside a human cell culture, for instance. Okay, so those are the antibiotics. Okay, so what differences do these antibiotics exploit? Okay, so we'll go through the list. The most common one, the very first antibiotic that was discovered, as you know, was penicillin. Penicillin comes from Penicillium chrysogenum, and it's not like we invented it. the fact that the fungi, a fungus called Penicillium chrysogenum, was already making this chemical that inhibited bacterial growth. Why would fungus be interested in making a chemical that kills bacteria? It's in order to eliminate competition for the same food. Remember that bacteria and fungi often grow on the same food sources. Bacteria divide a lot faster than fungi do, and so therefore, a competition between the two of them, the bacteria would always win if it was a fair competition, because the bacteria divide much faster than fungi do. The only way the fungi can survive is to create chemicals that inhibit the growth of bacteria. So the very first antibiotic that was discovered was penicillin. It turns out that penicillin inhibits the production of the peptidoglycan cell wall, and therefore these antibiotics, it turns out now that there's a whole family of antibiotics that specifically target the peptidoglycan cell wall that bacteria make. And so therefore these antibiotics will only be effective against gram-positive bacteria. So any of these antibiotics that target formation of the peptidoglycan cell wall will only kill gram-positive bacteria. They will not kill gram-negative bacteria. Now, when you get an antibiotic that only kills some types of bacteria but not others, we say that that is a narrow-spectrum antibiotic. A narrow-spectrum antibiotic, because it doesn't kill a lot of different types of bacteria. It only kills gram-positive bacteria. Right. And you'll see that there's some antibiotics that only inhibit the enzymes that make mycolic acid, and therefore that's a very narrow spectrum antibiotic because it will only kill the mycobacteria. Right. So you remember that we have Mycobacterium tuberculosis and Mycobacterium leprae. Those two will be killed by specific antibiotics that interfere with mycolic acid synthesis, but it will not harm other gram-negative bacteria. It won't harm most of the gram-positive bacteria. So those, again, would be an example of a narrow-spectrum antibiotic. All right, so starting from the top of the list, there are some antibiotics that target formation of the peptidoglycan cell wall. Those are narrow-spectrum antibiotics. They will only kill gram-positive bacteria. There are some antibiotics that target bacterial ribosomes. Remember that bacteria have something called a 70S ribosome, which is made of two subunits that are called 30S and 50S. I'm sure you remember why 30 and 50 do not add up to 70, and it's because these are based on Svedberg units. The S stands for Svedberg units, which are not additive. So humans have 80S ribosomes, bacteria have 70S that are made of two different ribosomal subunits, and so there are some chemicals that will specifically bind to the bacterial ribosome subunits and inhibit protein synthesis, right? So there's another class of antibiotics that does that. Those antibiotics tend to be broad spectrum antibiotics. They kill gram-negative, they kill gram-positive, they kill mycobacteria, they kill all of these, they will kill any bacteria that have 70S ribosomes, which is most of them. Interestingly, you remember that humans have mitochondria in their cells. The mitochondria do not have 70S ribosomes. They have 55S ribosomes because they're derived from archaebacteria. So we're in luck there. We're lucky there because you can take antibiotics that will kill the bacteria, but they will not kill the ribosomes. They will not inhibit the ribosomes inside the mitochondria. There is one antibiotic called kanamycin that will actually kill your ribosomes as well. So that is one of the ones that we don't use in humans. We use that one in the laboratory only. Okay, so anything that targets the bacterial 70S ribosome is pretty good because it's a very broad spectrum antibiotic. Okay, nevertheless, those antibiotics that target the ribosomes of a bacteria, they tend to be more toxic than the ones they tend to be more toxic to humans than the ones that target the peptidoglycan cell wall. All right, now bacterial enzymes, there's some antibiotics that will target the bacterial enzymes that make mycolic acid, and you only find mycolic acid in acid-fast bacteria like Mycobacterium tuberculosis and Mycobacterium leprae. Okay, and then there are some antibiotics that target and inhibit DNA replication enzymes. Specifically, when you're going to replicate your DNA, you have to, bacterial DNA is what's called supercoiled, which means that it's kind of, imagine taking a rubber band. You know, bacterial chromosomes are circular, so is a rubber band. Imagine taking a circular rubber band and you put it in your hands and you kind of rub it around between your two hands until it turns into a ball. What you've done to the rubber band is you have supercoiled it. Right, so bacterial DNA is supercoiled, right? And so if you want to replicate it, you have to unwind it before you can make a copy of it, right? And so there's an enzyme that participates in that called DNA gyrase. And humans have a DNA gyrase, and humans have other enzymes called DNA helicases that do this sort of thing. But there's a bacterial enzyme called bacterial DNA gyrase that can be inhibited by certain antibiotics. And so if you use those antibiotics, they will inhibit bacterial cell replication, bacterial cell division, by stopping the bacteria from being able to make a copy of its DNA. Okay, so DNA replication can be interfered with in bacteria, and those antibiotics tend to be broad spectrum antibiotics because it will interfere with the DNA gyrase from all bacteria. Okay, then there's some antibiotics that interfere with RNA polymerase enzymes in bacteria. Humans have 3 different RNA polymerase complexes, whereas bacteria have a single enzyme called RNA polymerase. And so we have humans and bacteria have different RNA polymerase systems. There are some antibiotics that will target the RNA polymerase that you find in bacteria. Those are broad spectrum antibiotics. And then finally, bacteria and humans both require a compound called folic acid to make a lot of their membranes and things. Humans get folic acid in their food. Bacteria generally do not. They generally make their own because they have enzymes to make their own. Humans lost the ability to make folic acid, but we can eat folic acid, whereas bacteria tend to make it. So there are some enzymes that make folic acid in bacteria, and there's some antibiotics that will inhibit those enzymes, thus preventing the bacteria from making folic acid. And therefore, this is also a broad spectrum antibiotic because all bacteria need folic acid. Right, so the antibiotics that interfere with peptidoglycan cell wall synthesis are narrow spectrum antibiotics. The ones that interfere with mycolic acid synthesis are narrow. The ones that interfere with ribosomes or with DNA replication or RNA polymerase or with folic acid synthesis, those antibiotics are all broad spectrum antibiotics. Right, now the ones that the antibiotics that interfere with cell wall production with peptidoglycan synthesis, they were originally, the original one was called penicillin. Since then, we've discovered that there are a whole bunch of different variations of penicillin. And if you put all of those variations of penicillin together, we call them the Penicillin family. They are, in fact, a family of antibiotics, all of which will only target gram-positive bacteria. So if you get infected with gram-negative bacteria, you need to use something else. If you're infected with gram-positive bacteria, it's actually preferable to use one of the penicillin family if it's not resistant to it, because the penicillin family tends to have very low toxicity to humans, right? So on the other hand, bacteria tend to build up a resistance to penicillin quite easily. So if you are infected with gram-positive bacteria, the odds are that you probably won't be able to use a penicillin, any members of the penicillin family to clear it up, because the last I've heard, something like 50% of the gram-positive bacteria in the world these days are now resistant to penicillin, so you have to use something else. Now, there is a man-made, artificially synthesized family of antibiotics that is very similar to the penicillin family. In other words, the organic chemists, the synthetic chemists got busy, and they used synthetic chemistry to build a whole family of antibiotics that are built similar to penicillin, but which are harder for the bacteria to develop a resistance to. And those are called the cephalosporins, and we're going to talk about those. All right, so example of antibiotics that inhibit mycolic acid synthesis that only affect acid-fast bacteria. Isoniazid is a good example of a chemical that will do that. Of course, that's very narrow spectrum because it will only stop acid-fast bacteria from building a mycolic acid cell wall. All right, how about antibiotics that inhibit the 70S ribosome? Example, the tetracyclines. Tetracyclines is a family of antibiotics, just as the penicillin family is a family. How about the antibiotics that inhibit nucleic acid synthesis by inhibiting bacterial DNA replication enzymes? Example, rifampicin is an example from the family that does that. There are some antibiotics that specifically punch holes in the bacterial plasma membrane, which means that they're very good, especially the outer plasma membrane, which means that they're very good at targeting gram-negative bacteria. So one example of an antibiotic that will do that is called polymyxin B, right? Polymyxin B. Now the problem with polymyxin B is that it's very toxic. And generally, if you have antibiotics that are toxic, that doesn't mean that you can't necessarily use them, but it does mean that you can't put them inside a person. You can use them as a topical anesthetic. T-O-P-I-C-A-L, topical. Topical means that you put it on the surface of the skin. So if you have a cut or a bruise, you can put polymyxin B onto the surface of the skin so that the bacteria won't infect the wound. But very little of the polymyxin B will get into the bloodstream and then make you sick. Whereas if you injected polymyxin B, that might kill you. All right, and then finally the bacterial antibiotics that inhibit folic acid synthesis. Example are a family of antibiotics called the sulfonamides. The sulfonamides, that's a family, right? So the tetracyclines is a family, the penicillins is a family, and the sulfonamides is a family. There are some other families that we'll talk about as well. So as I said, narrow spectrum antibiotics are very effective only against a few specific types of bacteria, whereas broad spectrum antibiotics are less effective, but they tend to affect many different types of bacteria. They tend to be more toxic to humans, which is why we haven't just given up on the narrow spectrum antibiotics. And every time somebody gets sick with a bacterial infection, why don't we just give them broad spectrum antibiotics? Why do we go through this? Why do we go through this big process of trying to figure out which bacteria is making you sick so that we can give you a specific antibiotic? Why don't we just give everyone broad spectrum antibiotics in the first place? Well, the reason is because they tend to be more toxic to humans. And so what used to happen if somebody got a bacterial infection, if they broke out in a fever and somebody looked in the blood and they saw bacteria in there or something like that, they would immediately put somebody on a course of broad spectrum antibiotics for a day or two, because it took a day or two to figure out which bacteria was infecting you. And then after 2 days, they do the streaking on a McConkey plate and a blood agar plate, and they put one McConkey plate in an anaerobic incubator and one in an aerobic incubator, and then they put the 2 blood agar plates in an anaerobic incubator and an aerobic incubator, and then they figure out exactly which bacteria is making you sick that way. Then they can give you a targeted narrow spectrum antibiotic. So that used to be the way it was done. That is not done very much anymore, or it's not done as much anymore, because these days people use PCR to figure out which bacteria you're infected with. Okay, so typically the treatment has been to give somebody broad spectrum antibiotics until you find out which bacterium is infecting them, and then you can give them a targeted narrow spectrum antibiotic. All right, so just to review, let's have a look at some of these ranges for the different. This is the bacterial spectrum. Don't worry about this over here on the right. These are drugs that will kill fungi and parasites, protozoa. You can see, for instance, that acyclovir is a chemical that inhibits viral synthesis, viral reproduction. It has no effect against helminths or protozoa or anything like that. Mefloquine or quinine is something that you would use to treat malaria, which is a protozoa, of course. Fungi, ketoconazole is what the ingredient that inhibits I believe that's the ingredient that inhibits ergosterol synthesis, but I'm not sure about that. It's been a while since I checked. And so these drugs over here on the right, I'm not going to ask you about them. So you don't have to worry about those, just for your own interest. All right, but you do have to worry about the ones over here that kill the bacteria on the left. Okay, so isocyanid is a very narrow spectrum antibiotic that only kills the mycobacteria, whereas streptomycin is a broader antibiotic that will kill mycobacteria and other gram-negative bacteria. Tetracycline, remember that tetracycline inhibits protein synthesis by interfering with ribosomes, and so that tends to be a broad spectrum antibiotic. And over here you have penicillin G, which is a very narrow spectrum antibiotic that will only kill gram-positive bacteria. Right, so typical example of a narrow spectrum antibiotic would be penicillin G. Typical example of a broad spectrum antibiotic would be tetracycline. So the penicillin family and the acid-fast bacteria and those that target just gram-negative bacteria like polymyxin B tend to be narrow. And then the broad spectrum antibiotics are the ones that inhibit folic acid synthesis or inhibit nucleic acid synthesis or inhibit protein synthesis by interfering with the ribosomes. All right, so where did we get these antibiotics from in the first place? In the beginning, we did not invent them. We discovered them because fungi were producing them. Right, now, the very first antibiotic was discovered by a scientist named Alexander Fleming, and he discovered it by accident because you know that fungal spores tend to be released in the summer when it's hot, and he was working in his lab. He was actually studying Staphylococcus aureus, and he had LB agar plates that he was growing, Staph aureus on, and some fungus got in. A fungal spore presumably floated in on the wind, and it contaminated one of his plates, and he noticed that where that fungal colony was, none of the Staph aureus was growing around it. It had a clearance zone, right? So there was a clearance zone around the Chrysogenum penicillium colony, and he suspected that the Chrysogenum penicillium fungus was producing some chemical that inhibited the growth of Staph aureus, and he investigated it and finally discovered penicillin. It was quite a very important discovery in 1928. So there are some antibiotics that are what are called natural antibiotics, and then because they come from penicillin or they come from fungi or other organisms, and then the synthetic chemists have made ones that are similar to that but harder for the bacteria to fight back against. And some of the antibiotics are what are called reformulated. Now, a formulation, you should be aware of the fact that the term formulation means putting them together in a different combination. Right? So a formula is what do you put together in what combination in order to make whatever it is you're trying to make. You use a formula. Okay, so formulations in the pharmaceutical industry, formulations means if you mix together two or more different drugs in a different solute or something, you can actually get those two drugs to do something that you didn't expect. Right? So different drugs will sometimes act synergistically. They will be better than either one of them alone if you mix them together in different formulations. So we have taken some of the early antibiotics and put them together in different formulations so that they can be used more effectively. Okay, so there's Alexander Fleming in the 1930s and 40s, showing him in his lab. So one of his plates got contaminated. So the plate had a Penicillium chrysogenum colony growing on it, and you can see that he had streaked Staphylococcus aureus on the plate, and the Staph aureus has a zone where it does not enter because there's what's known as a clearance zone where the bacteria didn't grow because the fungus was producing something that inhibited its growth. right? So we have since discovered a lot of other antibiotics from natural sources like fungi and even other bacteria. And then we've taken some of those and made artificial versions of them, and taken some of them and put them together in different combinations or formulations. Okay, now this is an interesting list. You should be aware of some of the sources of these antibiotics. So for instance, there's an antibiotic called bacitracin, which was originally isolated from a gram-positive bacteria, right? It was originally isolated from Bacillus subtilis, which is quite extraordinary. Why on earth would a bacteria make an antibiotic to kill other bacteria? Well, again, it's to eliminate competition. And another gram-positive bacteria produced polymyxin B, which is used as a topical anesthetic. Right. And then down here we have chloramphenicol. Chloramphenicol is commonly used as a laboratory antibiotic. Right. It was made by Streptomyces venezuela. Neomycin, we use that one in the lab all the time. It was produced by originally produced by Streptomyces fradiae. Streptomycin, another common one we use in the lab, originally derived from Streptomyces griseus, rather. Gentamicin, used in the lab all the time. Right, originally isolated from by the way, ascomycetes are a special type of bacteria, right? So you know the special type of bacteria, ascomycetes, or fungus rather. We studied them before, actinomycetes, right? So these are some of the. It's actually actinomycetes are a special type of microbe, which sort of halfway fits into the bacterial category, but we won't go into that. And then, of course, you know that penicillin was originally discovered from Penicillium chrysogenum, right? Okay, now modern day research is kind of interesting because the bacteria are kind of catching up with us. We had a beautiful golden age from about 1930 until about the year 2000, where whenever you got a bacterial infection, we could easily cure it by giving you bacterial antibiotics. But the bacteria have been catching up to us by developing various forms of resistance. And so we are rapidly running out of antibiotics that we can use to kill bacterial infections. And so modern research, the new research, has moved on to studies of naturally occurring antimicrobial peptides. So if you remember from your first year classes, peptides are short proteins, right? And it turns out that many animals, many exotic animals, and including some humans, make interesting short peptides that have the capacity to kill bacteria. And so now that we've sequenced the genomes of all sorts of animals and things, we're rapidly looking through the genomes of animals that produce antimicrobial peptides, and using those doing research into those as an alternative to antibiotic chemicals. Now, any of you students that are interested in going into microbiology and you're interested in going into UBC, you should know that UBC, right here in British Columbia, we have two of the most famous microbiologists in the world, one of whom is a guy named Robert Bob Hancock. I had him as a teacher when I was at UBC. I don't know if he's still working. He's probably retired by now, but he taught me. And he's a world famous microbiologist who invented and discovered these antimicrobial peptides. And it was quite interesting. He discovered these, and he discovered antimicrobial peptides, and then he discovered another type of antibiotic killing peptide called a cationic peptide that also kills bacteria. And he actually donated, rather than patenting and making a lot of money off of the antimicrobial peptides, he donated them all to the United Nations, donated the knowledge to the World Health Organization, so that everyone in the world can do research on these things that can be used to kill bacteria free of charge, because Bob Hancock was generous enough to donate them to the World Health Organization. He then founded a company with the other thing, the cationic peptides, and he made some money that way. So he's kind of an interesting guy, originally from Australia, but has spent his entire career at UBC. He donated this huge field of research into antimicrobial peptides to the United Nations and to the World Health Organization. So he's a big hero in everybody's eyes for that. And then, but he wasn't giving up everything. He kept some of it for himself to make money for himself, and that's fine, right? So anyway, he was. As some of you may know, we don't have knighthoods in Canada, but here he is getting the Order of Canada medal. This is his official Order of Canada picture. Canada kind of gives away these things called, there's an award that you can get called the Order of BC if you've done things that are of public service to BC. And then if you've done things that are of great service to Canada, you get the Order of Canada. So here's Bob Hancock's picture as he's got the Order of Canada medal. Anyway, he's a very famous microbiologist who is a professor at the UBC Department of Microbiology. Some of you might someday want to go and try and get into his lab to do work as a graduate student or something like that. That would be a very nice, interesting thing to do. And here's another famous UBC microbiologist who's also got, as you can see, he's also been awarded the Order of Canada. His name is Brett Finlay. He is also a world famous microbiologist, and I'll explain why as we go through this. But one of the problems with antimicrobial resistance is that one of the things that farmers do is they put antibiotics into chicken feed and into cattle feed in order to kill off the symbiotic bacteria in the cattle and the chicken guts so that the chickens and the cows gain weight and get bigger faster. It's a way of making profits. This is contributing to antibiotic resistance, because the more you use antibiotics, the more of them become resistant, and then the antibiotics lose their effectiveness. And so for decades, ecologists and other biologists and people who are concerned about the future of antibiotics and even the future of the environment have been telling Canadian farmers, look, you shouldn't be putting antibiotics into the chicken feed and the cow food, because that's going to cause that should be illegal because it's contributing to antibiotic resistance in the world. And the farmers are saying, okay, well, we don't want to do anything dangerous, but you realize that if you forbid us from doing that, the American farmers in the United States are not going to stop, and then they'll just drive us all out of business. And then, or the farmers in the European Union will they're not going to stop. And so if the Canadian government stops us from putting antibiotics into our chicken feed and cattle feed, you're just going to succeed in driving only the Canadian farmers out of business. Okay, so that's not a very nice attitude, but it's an understandable attitude. And so, in my opinion, the mark of a good scientist and a good public scientist, somebody like Brett Finlay, is to understand that not all of the, it would be great if every farmer in the world stopped putting antibiotics into their food, and so we wouldn't have to worry about this. But realistically, that's not going to happen for reasons of commerce. And so a good scientist looks for a practical solution to these problems. A good scientist will look for a realistic solution. And so what Brett Finlay did was he figured out, okay, we're not going to stop the Canadian farmers from putting antibiotics into their feeds. Instead, why don't we vaccinate the cows and the chickens against bacteria so that we'll kill the bacteria in their gut that way? And then that's an alternative, right? So we're going to, as an alternative to giving cows and chickens antibiotics, Brett Finlay started this program of vaccinating them instead. And also he started this program of vaccinating cattle against enterohemorrhagic E. coli and other, the pathogenic forms of E. coli that make people sick. It was Brett Finlay's idea to just simply, instead of trying to cure people when they eat EHEC or enterohemorrhagic E. coli, why not vaccinate the cows against the virulent strains of E. coli so that you never end up having hamburger that contains EHEC E. coli. Right? So that was a very good suggestion of his. And so Brett Finlay is a very famous microbiologist in Canada. And he's at UBC as well. He's in the UBC Department of Microbiology, and so maybe some of you might be interested in going and working in his lab someday. All right, so let's go back to the Penicillin family. So let's take a look at the antibiotics that inhibit cell wall synthesis. They inhibit the formation of peptidoglycan cell walls. Okay, so as you already know, gram-positive bacteria have a thick layer of peptidoglycan on the outside of the cell of the plasma membrane. Gram-negative bacteria have two cell membranes. They have an outer plasma membrane and an inner plasma membrane. The space in between is referred to as the periplasmic space, and it's filled with a fluid called the periplasm, and it contains a small amount of peptidoglycan. But if you interfere with that formation of that, the antibiotic has trouble getting through the outer cell plasma membrane. So basically, the Penicillin family of antibiotics will interfere with the peptidoglycan cell wall formation of gram-positive bacteria, but not gram-negative bacteria. Okay, so the gram-positive bacteria, they have a peptidoglycan cell wall. Right, so penicillin is a chemical that actually resembles peptidoglycan, right? And it has a central feature called a beta-lactam ring. That's very important that you know what that is, right? A beta-lactam ring. And so any of the antibiotics that inhibit the formation of the peptidoglycan cell wall are members of what's known as the beta-lactam family of drugs. Right? Now, if you put beta-lactam, if you put any penicillin with the beta-lactam ring, if you have it around while the bacteria is building its cell wall, the penicillin or any drug that has the beta-lactam ring will accidentally be incorporated into the cell wall, and it will cause the cell wall to fall apart. Okay, so you see down here I've written triangular brick analogy. Okay, just let me give you this analogy. Suppose you have a bricklayer who's putting together a brick wall, and all of the bricks that he's putting into the brick wall are square. What if you secretly, suppose he was a blind bricklayer and he couldn't see the shape of the bricks. What if you threw a bunch of triangular shaped bricks into his pile of bricks so that every third or fourth brick that he puts into the wall is actually a triangular brick instead of a square brick? The wall would just collapse. Right? And so that's exactly what happens with the peptidoglycan cell wall for gram-positive bacteria if beta-lactam containing compounds are around. The beta-lactam ring, the beta-lactam compound is accidentally put into the wall, and then it falls apart. Now, one of the interesting things about this is that bacteria, gram-positive bacteria, are only building the cell wall when they're dividing. Right? So I asked you a question earlier on about when we're talking about bacterial growth curves. You know, you have the inoculation phase, then you have the exponential phase, then you have the stationary phase, then you have the death phase, right? And I asked you, well, at which of these phases is the bacteria, if it's a gram-positive bacteria that's being killed by penicillin, at which of those phases would it be most vulnerable? Right? And the answer is it's most vulnerable during the logarithmic growth phase, the log phase, which is a period of exponential growth. Right? It's only vulnerable when you have the most cell division going on, which happens during the log phase. Once you get into the stationary phase and the bacteria are not dividing anymore, they're just sitting there and getting bigger, then it's not such a big deal. The penicillin and penicillin family antibiotics cannot kill bacteria, gram-positive bacteria, when they are in the stationary phase, only when they're in the log phase, when they're actively dividing. Okay, so here we see what happens. On the top we have normal gram-positive bacteria. On the bottom, this is what happens to bacteria when there is a beta-lactam compound around. And what actually happens is that the cell wall kind of helps gram-positive bacteria from bursting if the environment is slightly hypotonic. So you know that if you put a bacterial cell in a hypotonic environment, the water will go into the bacterial cell, it will swell up and possibly burst, and the bacterial cell wall kind of prevents that from happening. Right? So this is how the penicillin family of antibiotics kills these bacteria. If they interfere with construction of the peptidoglycan cell wall, then the bacterial cell is much more vulnerable to lysis, plasmolysis, by being in a hypotonic environment. All right, now let's talk for a second about antibiotic resistance. So there are many bacteria that are resistant to penicillin, and here's how. So we have a chemical called penicillin that contains the beta-lactam ring, and you know that there are enzymes around whose job it is to hydrolyze different types of polysaccharides, right? So there's an enzyme around called the beta-lactamase enzyme that is able to hydrolyze the beta-lactam ring, right? So what happens is this gene, by the way, is carried on a plasmid, which bacteria can pass back and forth to each other through either conjugation or just simple transformation. And this enzyme that's on a plasmid, the beta-lactamase enzyme gene, the beta-lactamase gene, is the bacterial cell will produce beta-lactamase enzyme and send it out into the environment. And the beta-lactamase that's sent out into the environment will cut up and deactivate the penicillin before it has a chance to be incorporated into the bacterial cell wall. So that's how the resistance works. We're trying to kill the gram-positive bacteria by putting these triangular bricks into the environment, the beta-lactam drugs. And the bacteria is defending itself by sending out this enzyme to chop up the beta-lactam compound before it gets incorporated into the cell wall. And as I said, the gene is encoded, it's carried on a plasmid, which these bacteria can pass around to each other. So here we have the beta-lactam ring, right? So that's the beta-lactam ring right there. This is a slightly different compound down here below, but that's the beta-lactam ring. So that is the spot that beta-lactamase chops. It chops these things in half and deactivates them. Okay, so the beta-lactamase enzyme is encoded by a gene called BLAF, or beta-lactamase, right? So there's the lactam ring. Beta-lactamase will chop it like that and basically deactivate it so it doesn't get incorporated into the cell wall. Typically what happens is if somebody is sick because they're infected with gram-positive bacteria, you give them penicillin, and 99.9% of the bacteria that's infecting them does not have the beta-lactamase gene, but 0.1% of them do. So what happens is you take the course of antibiotics, and the antibiotics will kill off all of the bacteria except for the ones that are resistant, and then you'll be healthy for a while. And if you're lucky, your immune system will clobber and kill off the rest of the bacteria that are resistant. If your immune system doesn't kill off the resistant bacteria, they will repopulate your body, and then you're in real trouble. Right. So the use of antibiotics is what is, so notice that before we gave that person the antibiotics, they had a body full of bacteria, and only a few of those bacteria were resistant to the penicillin. But after we gave them a course of penicillin, then their body was repopulated, and now they've got a lot of bacteria that are resistant to penicillin, and that's where the penicillin-resistant microbes, bacteria, are coming from. They get increased in number every time we use antibiotics. Okay, so superbugs is what we call antibiotic-resistant bacteria. These are antibiotic-resistant bacteria. In this case, the superbug is resistant to any of the beta-lactamase family of antibiotics. The beta-lactamase gene is carried on a plasmid. It's called the BLA gene. The plasmid can be passed around. There are many other examples of different antibiotic resistance mechanisms that are carried where the genes that encode the resistance are carried on plasmids, and so they can be passed from one bacteria to another, or they can, if you already have the gene, you're resistant to the bacteria, and then you can repopulate the area after all of your cousins that didn't have the resistance have been killed off. Okay, so this alarming graph that I showed you before is demonstrating the rise of methicillin-resistant Staphylococcus aureus. So you do definitely, at the end of this course, you do need to know what MRSA stands for. MRSA stands for methicillin. Methicillin is an antibiotic related to penicillin. It's usually used as a last resort treatment against Staphylococcus aureus. Do you remember whether Staphylococcus aureus is gram-negative or gram-positive? Of course, Staphylococcus aureus is a gram-positive cocci. And generally, if somebody has a staphylococcal infection, you try different antibiotics. You try the easy ones like penicillin or something. And if none of those work, as sort of as a second to last resort, you try methicillin. But these days, a lot of the Staph aureus is resistant to methicillin. And the last resort that we have, the last drug that you would typically give somebody that's resistant, has methicillin-resistant Staph aureus, is you would give them a drug called vancomycin, which is a very toxic drug. Right? And so the vancomycin, if the person is sick because they already sick enough because they've got the Staph aureus infection, if you give them vancomycin, the vancomycin may cure them or it may kill them. So we don't like being in a position where a lot of the Staph aureus in the world is already resistant to methicillin. And if you find somebody who's sick with MRSA and they're very sick because of the Staph aureus infection, the last resort you have is the vancomycin, which is very toxic to humans. Okay, so this graph shows you that way back in 1989, about 2% of the Staph aureus that people, every time somebody gets a Staph aureus infection, they go to the hospital, they test to see if it's resistant to methicillin. Back in 1989, only about 2% of the Staph aureus infections were resistant to methicillin. Look at it in 2002, it's nearly 40%. I don't know what it is now. I think it's up around 60% or something like that. And when it gets to 100%, then we're really in trouble, aren't we? And basically anybody who gets infected, you know, this is like playing Russian roulette, you know, that game where you put a bullet in a revolver and then you spin it and put it to your head and pull the trigger. And then if you put one bullet in and you have one chamber that has a bullet and you have 5 that are empty, your chances of living are 5 out of 6, right? So here, if you get infected with infected with a Staph aureus infection, your odds of methicillin being able to cure it in 2002 were only 4 in 10. Right now, it's the odds, or 6 in 10 rather. Right now, the odds in 2020 are something like only 4 in 10 that you can cure the disease with methicillin. So this is a problem. This is a very serious problem for the human race. The fact that the antibiotics are losing their effectiveness and the bacteria are catching up to us, and they're fighting back. And so that's why it's so important. What the things like Bob Hancock discovered, the antimicrobial peptides, that's why I think finding new ways to kill bacteria are so important. Okay, so how do you combat these superbugs? Okay, how do we, you know, in the absence of new things to kill bacteria, what can we do to slow down the rise of superbugs? Okay, so number one, you should only take antibiotics when they're needed. Right, so if you all know this, but most of the other members of the public don't know this, and so it's your duty now to explain this to everybody in order to save the human race. So you should save the human race by explaining to as many people as possible that you do not take antibiotics for a cold, because a cold is caused by a virus. Now, sometimes if you catch a really bad cold or a really bad flu, you will get a bacterial infection of the lungs, which is secondary to the original viral infection, and therefore you would have to take antibiotics. And there are some doctors around, and even there are many places in the world where they sell antibiotics over the counter. You don't need a prescription to buy antibiotics. And so some people think, well, okay, I realize the fact that colds and flus are not caused by bacteria, but I'm going to take antibiotics anyway in order to preemptively prevent lung pneumonia, bacterial pneumonia, secondary to the flu or secondary to a bad cold. Right? So you shouldn't be doing that. Nobody should ever be doing that, because that contributes to the rise of superbugs. Okay, that encourages the development of superbugs. Okay, so don't take antibiotics when they're not needed. As I said, there are some places in the world most places in the world you need a prescription, and the doctor is not supposed to give you a prescription for antibiotics just for a cold or the flu, right? And so, so there's some places in the world where they sell antibiotics over the counter. That needs to be stopped. Otherwise, the survival of the human race is in jeopardy. But in addition to that, even in places like Canada, where you have the doctor has to prescribe antibiotics, I've literally seen this happen, where somebody will come into a doctor and say, I've got a cold, I want antibiotics, and then the doctor says, you don't need antibiotics for a cold, get out of my office, right? So then they leave and they go to another doctor and they say, I want antibiotics, and the second doctor says, you don't need antibiotics for a cold, and then they'll stay and argue, and the doctor will try and explain it to them, and they'll leave and they'll go to another doctor, and finally they'll find a doctor who is willing to just write them a prescription for antibiotics just to get them out of their office. Just to stop them from being annoyed by this person demanding antibiotics. And that is highly irresponsible. That's really that's a professional irresponsibility on the part of the doctor who does that, but there are doctors who do that, I'm sorry to say. So that is one thing that needs to be stopped. Okay, now finally, if you actually get a bacterial infection so that you do need an antibiotic, a course of antibiotics, when you're given a prescription for antibiotics, you have to take the whole thing. Right? So let's say they give you 60 tablets, and they say take 3 a day for 3 weeks or something like that. And then you feel better after 2 weeks, and then you say, well, I won't take all of these, I'll save them for the next time, and then I don't have to go and buy them again. Or if I feel myself getting sick, I'll take them next time. No, you can't do that, because what you've done is you've stopped your war on the antibiotics. You've stopped the war on the bacteria before they were all dead. Right? So if you the course of antibiotics, 10 days, 3 times a day, I'll give you, 90 tablets. That has been calculated to kill as many of the bacteria as is possible. And if you call off your army before the other army is destroyed, they'll come back. Right? So another thing that you should never be doing is not taking the whole course of antibiotics. If you're prescribed a course of antibiotics, you must take the whole course. You must take all of the pills as prescribed. Right, so no saving half of the antibiotics for later in case you feel sick in the future and you don't have to pay for another bottle. You have to take them all, because if you don't, then you're encouraging the survival of superbugs. All right, so what is a formulation? A formulation is where you mix different drugs together to get better effects if they're synergistic, or to increase longevity in the body of the drug. Okay, so mixing penicillin or members of the penicillin family with chemicals can increase its longevity. The word longevity means how long does it last? How long does it last? Longevity. Okay, so if you mix, there are also some formulations where you can mix penicillin with chemicals that inhibit beta-lactamase. Right, that's very clever. So we're trying to kill the bacteria by putting beta-lactam into their environment. They fight back by sending out beta-lactamase enzyme, and then we send in a chemical that inhibits the beta-lactamase enzyme. And so this is a very complicated battle, but that's another thing that we can do. All right, so the other final thing is that penicillin, the original penicillin that was discovered by Alexander Fleming, is now called penicillin G, and that's the natural form of penicillin, and it had to be injected into the blood because if you took it as a pill, the digestive enzymes in the stomach would simply tear it apart. Right, so it would not survive. The original penicillin G you could not take orally. The word oral means by mouth. And so the original penicillin you could not take orally because it would be destroyed in the intestines. It would be broken down by various enzymes in the intestines. So what they've done now is they've used synthetic chemistry to modify the original penicillin G a little bit here and there to create other penicillins that can be taken orally. All right, so now let's talk about the penicillin family. Right, and we'll start with the natural penicillins. So this is the original penicillin plus some minor modifications of the original penicillin plus some different formulations of the original penicillin. And then we'll move on and talk about the cephalosporins. The cephalosporins are an artificially made version of penicillins that are harder for the beta-lactamase enzyme to break down. Okay, so as we said, the active part of the original penicillin is the beta-lactam ring, and it gets incorporated into the cell walls of gram-positive bacteria. Right? Bacterial resistance, they make an enzyme called beta-lactamase enzyme, which cuts up the beta-lactam ring and destroys it. Okay, so penicillin G, that was the original penicillin derived from Penicillium chrysogenum, the fungus. It had to be injected into the blood. It did not, once it was injected in the blood, it decomposed fairly quickly, so you had to keep on injecting it. Right? So then they made a new formulation, a chemical formulation, which is called procaine penicillin, which is again injected into the body. It has to be injected, but it increases how long the penicillin lasts in the bloodstream. Right? So penicillin G has to be injected. That's the original. Procaine penicillin lasts longer in the blood, but it still has to be injected. There was another formulation, another minor modification of penicillin called benzathine penicillin, which again you have to inject, but it's meant to be injected intramuscularly into the muscles where it lasts even longer than if you inject it into the blood. And then finally, the synthetic chemists modified the original penicillin G a little bit to create something called penicillin V, which can be taken by mouth, orally. Okay, so the original pharmaceutical companies grew up large amounts of Penicillium chrysogenum in these large fermenters, and then you can take the penicillin, you can extract penicillin G, and then you can take the penicillin G and use it, or you can modify the penicillin G into penicillin V or other things using synthetic chemistry. All right, now this graph is interesting and important. So you inject penicillin G into the bloodstream, it reaches a very high concentration in the bloodstream, and then it decomposes very quickly and it's gone, and then you have to inject it again, right? Okay, then don't worry about this one for a minute. Then we have the procaine penicillin, which has longer longevity, and then we have the benzathine penicillin, which if you inject it into the muscles, it lasts for a very long time, for 30 hours. Right? And then finally, the oral version, which is called penicillin V, it's a variation of penicillin G. It can be taken by mouth, but you have to take it a few times a day in order to keep it in your bloodstream. Okay, so there's Propen G, that stands for procaine penicillin G, which needs to be injected. It says so right on the bottle. Okay, so the modification that the synthetic chemists made to the penicillin G was just this. Apparently, just by adding the extra oxygen there, that prevents enzymes in the intestine from breaking it down, which is what allows it to survive a trip through the stomach and the intestine. All right, now the cephalosporins. These are the various synthetic versions of the penicillin family. So oxacillin is a synthetic chemistry version of the penicillin, which is harder to break the beta-lactam ring. Methicillin, which you've heard of by now, is a very strong antibiotic. It's used as the second to last resort against Staphylococcus aureus infections. Many of the Staph aureus are now resistant to it, so we have to use vancomycin, which is even worse. Amoxicillin is generally used topically because it's a bit toxic. And then finally, a synthetic version of penicillin, which is called ampicillin, is very toxic to humans, and so we only use that in the lab when we're doing tissue culturing. We sometimes put it on a plasmid. If you're using bacteria to make a foreign protein, you put the foreign protein gene into a plasmid, and then the plasmid also contains the beta-lactamase gene, so that you can put the plasmid into the bacteria, and then you grow them in a culture that contains ampicillin. And so that's a way of preventing your bacterial culture from being contaminated by just regular old E. coli that gets contaminated into the culture. So you'll learn all about that if you take biology 200, and you talk about using bacteria to make synthetic peptides and synthetic compounds. All right, so this is just some of the modifications. You don't have to memorize the modifications. So there's the modification made to penicillin G to create penicillin V. And then there's some other modifications to these areas here to make the other forms of synthetic penicillins. Notice that they all have the beta-lactam ring though. Okay, now here's some formulations of penicillins and cephalospore and synthetic, natural versus synthetic penicillins, different formulations. Okay, now clavulanic acid is a chemical that inhibits beta-lactamase, right? So if you mix that in with a penicillin or any member of the penicillin family or the natural versus synthetic penicillins, then you will increase the effectiveness because clavulanic acid is inhibiting the beta-lactamase, and therefore it can still the whatever penicillin family you put in there can still kill the bacteria. Okay, so one of the synthetic penicillins, amoxicillin, which we just learned about, if you mix that with clavulanic acid, it's sold under the brand name Augmentin because you've augmented the effectiveness of the amoxicillin, right? So the brand name Augmentin is a mixture of amoxicillin and clavulanic acid, right? And that is used to increase the potency by inhibiting the beta-lactamase enzyme. So there it is. Okay, now some of the problems with the penicillin family, by the way, that last slide that I showed you were not the cephalosporins, they're coming up, but some of the problems with the natural penicillins as well as the synthetic penicillins is that many of them, first of all, they have a narrow spectrum. Many of them have side effects. They'll make you nauseous, right? That's why sometimes they'll say take it with food because you feel very nauseous if you take it without food. And then many of them are allergenic. So many people are allergic to penicillin. And so if you give somebody like that penicillin or even any of the penicillin family, they may go into anaphylactic shock. So you know that anaphylactic shock is where you have the capillaries become leaky and a lot of the serum of your blood leaks out into the soft tissues. That causes a rapid drop in blood pressure, which could lead to a heart attack, or you'll faint, or possibly have a heart attack because the blood pressure is so low that the blood is not carrying oxygen around to the brain and the heart. Okay, so you should always be tested for allergy to penicillin and the penicillinase family. And if you're allergic, you need to wear a medical alert bracelet, right? Because if you get an infection and you go into a coma or something and they're thinking about killing the bacteria with penicillin or something, they don't want to give you penicillin. Okay, so if you can, you should, next time you're doing a checkup at your doctor, you're going into a doctor's office for another reason or whatever, ask them if you're allergic to penicillin, and if they don't know, if it's not in your medical record, maybe they could do the test. And so this is anaphylactic shock. Some of you have seen this slide before. The serum leaks out of the blood into the soft tissues and it interferes with various body functions. If you go into anaphylactic shock, you have to have a shot of adrenaline. These EpiPens contain adrenaline for people that are allergic to peanuts. If you eat peanuts, you go into anaphylactic shock, and therefore you have to have a shot of adrenaline. Okay, so this is how you get tested for various things, various allergies. They just put this thing on your back, and then you're tested for reactions to various things that many people are allergic to, including penicillin. Okay, so the cephalosporins are the artificial versions of penicillin. So they're not modified natural versions. The last two slides I showed you were of the natural penicillins. These are the artificial penicillins, the cephalosporins. So the cephalosporins are completely synthetic versions of penicillin. It turns out that these have been built by synthetic chemists, and it's harder to break the beta-lactam ring. They have greater longevity. They are less allergenic, so they cause anaphylaxis less. Okay, so example, one of the cephalosporins is called Kefalex, which is sold under the brand name Keflex. Right, so be aware that Keflex is a brand name for Kefalexin, which is one of the cephalosporins, and the cephalosporins are completely synthetic versions of penicillin. Okay, so we have the this is what the cephalosporin, this is the main difference between cephalosporins and natural penicillins here. All right, now, vancomycin and bacitracin are two other things that will inhibit cell wall synthesis through other mechanisms. Right? Vancomycin is a short peptide, a glycopeptide that inhibits an enzyme that's involved in making the peptidoglycan. It is very toxic to humans, right? And so vancomycin A is definitely a last resort against methicillin-resistant Staph aureus. So if you get Staph aureus, they might try a routine natural penicillin or a cephalosporin. If that doesn't work, they'll try methicillin. If the methicillin doesn't work, they'll move to vancomycin, which is quite bad for you. And bacitracin, remember, was originally isolated from a bacillus species, right? And it inhibits peptidoglycan synthesis, but it is so toxic that it is generally used topically. Topical means as an ointment, as a cream, right? So polysporin contains bacitracin in it. So you can buy this cream that you're supposed to put on cuts and scrapes called polysporin. One of the ingredients of polysporin is bacitracin, which is why it's only used topically as a surface antibiotic. Okay, there's vancomycin. There's bacitracin ointment, and some of the bacitracins go under the brand name polysporin. All right, so we have the penicillins and the synthetic penicillins, which are modified versions. Those include amoxicillin, methicillin, and so on. We have the cephalosporins like Keflex. Kefalexin is an artificial penicillin. And then we have vancomycin and bacitracin. All of these are narrow spectrum because they only treat gram-positive bacteria, and some of them cause anaphylaxis. Right, so these all contain the beta-lactam ring, which the bacteria can fight back against by using beta-lactamase enzyme. Right, these have a modified beta-lactam ring, which is much more difficult for beta-lactamase to cut. So these are safe from some of the So if it turns out you're allergic, you're infected with a bacteria that's resistant to penicillin or the synthetic variations of Penicillin family, you can try cephalosporins because you're more likely to be successful there. Okay, what about the mycolic acid synthesis inhibitors that will stop any of the acid-fast bacteria? Okay, so Mycobacterium tuberculosis and Mycobacterium leprae produce mycolic acid. There are two drugs, isoniazid and ethambutymol are two chemicals that inhibit mycolic acid synthesis, and they are narrow spectrum because they will only kill mycolic acid bacterium. However, if you have leprosy or tuberculosis, you can use those two drugs very effectively, but people also if you have leprosy or tuberculosis, people are often treated for leprosy or tuberculosis using two broad spectrum drugs, streptomycin or rifampin. Right? So rifampin inhibits replication of the DNA, inhibits RNA polymerase in bacteria, and streptomycin is a protein synthesis inhibitor. Right? So these are the four drugs that are typically used to treat tuberculosis and leprosy. Okay, what about inhibition of protein synthesis by inhibiting ribosomes or ribosome subunits? Right, so remember that bacteria have a 70S ribosome, humans have an 80S ribosome. The bacterial 70S ribosome is made of a 30S subunit and a 50S subunit. Okay, so now there are several different antibiotics that will target either the 50S or the 70S subunit. Okay, so the aminoglycoside family, this is a family. These are bactericidal because they inhibit the 30S subunit irreversibly, right? So they are irreversible inhibitors of the 30S subunit. So they are bactericidal. They will actually kill the bacteria. You notice that underneath we have the tetracycline family and the macrolide family. They are both they're reversible, and so they're simply bacteriostatic. Right? Okay, so in other words, when they wear off, the bacteria can come back and start dividing again. So typically the use of tetracyclines and macrolides is limited. Okay, but anyway, so going back to the aminoglycosides, right? So they irreversibly bind to the 30S subunit and inactivate protein synthesis permanently. Unfortunately, they have glycoside family of antibiotics have very strong side effects, including kidney damage and hearing loss. I think I said liver before, but I meant kidney. Right? So kidney damage, right, and hearing loss, and so we only use those in the lab for tissue cultured cells and to maintain plasmids if you're trying to produce insulin in a strain of E. coli or something like that. Right? So that's what you would use the aminoglycosides for. So two examples of the aminoglycosides are kanamycin and neomycin. You should memorize those two antibiotics. Okay, the tetracycline family, there's a number of them. We're not going to memorize any specific ones. They tend to be bacteriostatic. They bind reversibly to the 30S subunit. And so, if you have a minor infection, in some cases you can reduce the number of bacteria present with a tetracycline, and then your immune system can take over and kill the rest of them. More commonly, if you have a fish tank and you have sick fish, or if you have a bird like a budgie or a parrot and the parrot gets sick, you go to the veterinarian and they give you some antibiotics to put in the fish's water, or they give you some antibiotics to put into the bird's water. It's usually a tetracycline. Right, so it's usually tetracycline, and tetracyclines, by the way, are yellow in color. So they give you this little pill at the pet food store, and you put it into the bird's water and it turns yellow. That's the color that tetracycline is. Okay, then we have the macrolides. Again, they're bacteriostatic, meaning that they bind reversibly to the 50S subunit this time, right, to the 50S. They're often used for oral use. In other words, they put them in, if you have a sore throat because you have just because you've been yelling or something like that, this wouldn't do to cure a strep throat, or it might, but they put them in, those throat sprays that are meant to cure your throat of a throat infection, or you take it in a, it's orange flavored, and you take it in a little cup to cure a minor bacterial infection. That's usually one of the macrolides, and the best example is erythromycin. Right, so they're not terribly effective at killing bacteria because they are bacteriostatic, not bactericidal, but they're also relatively harmless. Right, so they're prescribed for a lot of things. You can buy most of the macrolides over the counter. So the throat sprays for a sore throat and the little things that you take, sometimes you suck on these lozenges and they contain macrolides. They're relatively harmless. Okay, chloramphenicol. It reversibly binds to the 50S ribosomal subunit. It's cheap, but it's somewhat toxic, and so we don't use it in humans. It has been used for decades in cattle feed, which is a bad thing because then the cattle feed are developing resistance to that one. So that causes various other problems. They're trying to phase that out now through vaccinating cattle instead of giving them antibiotics. Okay, so as I said, if you have a sick bird, it's usually bird antibiotics that you put into the bird's water. Those are usually tetracycline. Here we're showing some eye drops. So if you have an eye infection, a minor eye infection of some sort, usually there are erythromycin or other macrolides in the drops or in the throat sprays. And chloramphenicol put in cattle feed. As I said, Brett Fenley at UBC has started this program to stop them from doing that, and as an alternative, they're vaccinating cattle against bacteria instead of giving them antibiotics. Okay, what about inhibition of nucleic acid synthesis? Okay, so these bacteria have their own DNA and RNA replication enzymes. These tend to be broad spectrum, these antibiotics. So the quinolones, any of the quinolones will inhibit the DNA gyrase enzyme that I mentioned earlier, which makes it impossible for the bacteria to make a copy of its DNA. And so this interferes with DNA replication. Now, this is an interesting question. There's sometimes if you take two different types of antibiotics together, they will work synergistically, meaning that they attack two different mechanisms to do with the bacterial cell. On the other hand, sometimes if you take two different bacteria together, they work against each other. They're antagonistic to one another. And the quinolones prevent cell division. And I just mentioned to you a minute ago that the penicillin family can only kill bacteria during cell division, right? So it stands to reason that if you took quinolones at the same time with penicillin, the quinolone would essentially the quinolone antibiotic would essentially neutralize the penicillin family, right? So those are two types of antibiotics that you would typically not take at the same time. But there are others that you can take and they have synergistic effects, right? So anyway, the quinolones, they inhibit DNA gyrase activity. They're sometimes referred to as fluoroquinolones because many of them have a fluoride group as well. And they contain fluoride. Example, ciprofloxacin, right? So ciprofloxacin is sold under the brand name Cipro, right? And it is a broad spectrum antibiotic, ciprofloxacin, that will inhibit the DNA replication, stops the bacteria from replicating. Another one is rifampicin, and it inhibits the RNA polymerase, so it can't make, in a way that inhibits protein synthesis as well. So you can see that if you took tetracyclines together with rifampicin, for instance, they would be synergistic because they're probably they would be a little bit synergistic or perhaps neutral because they're both inhibiting the same process, protein synthesis. It's just that one of the antibiotics is preventing messenger RNA from being made, and the other one is preventing the messenger RNA from being translated. So anyway, rifampicin inhibits the RNA polymerase enzyme, and one of them goes under the brand name Remactin, right? So rifampicin is another broad spectrum, and it sells under the brand name Remactin. All right, so there's the gyrase unwinding the DNA so that you can replicate it. If you inhibit that, then you can't make a copy of the DNA and the cell cycle is stopped. Right? So there's ciprofloxacin tablets. Right? And here's RNA polymerase. You know all about that by now. Remactin. Okay, and finally there's some things that punch holes in the bacterial plasma membrane. Okay, so gram-positive bacteria tend to be less affected because the cell wall stabilizes. If you punch holes in the plasma membrane, it has less of an effect if there's a cell wall to help stabilize the inner membrane, despite the fact that it has holes punched in it. Okay, so polymyxin B, originally isolated from Bacillus polymixa. It's a natural catatonic peptide. I mentioned that Bob Hancock at UBC is studying catatonic peptides. It breaks holes in the outer plasma membrane of gram-negative bacteria. So this is it's a narrow spectrum antibiotic in the sense that it only affects gram-negative bacteria. It tends to be used, it's toxic, so it tends to be used only as a cream, a topical antibiotic. So it's topically used only, topical use only. It's in polysporin, right, which is an ointment that you can buy. Right, so polysporin, you should know this, polysporin contains polymyxin B, neomycin, and bacitracin. Right, so bacitracin is the weakest of the three ingredients. Right, neomycin is strong but toxic, so you wouldn't want to put that inside your body, but it's pretty good at inhibiting bacterial protein synthesis in the bacterial cell outside the body. And then polymyxin B punches holes in the cell membrane of gram-negative bacteria. Right, so probably these three are toxic, but as long as you use them in a topical cream, they're actually working synergistically, aren't they? Right, so you have one antibiotic that's punching holes in the bacterial plasma membrane, another one that's messing around and stopping the activity of the ribosomes, and another one that's using another using a mechanism that's different. So the three of them together will be synergistic. So there's polysporin, and now you know what's in it. And as I said, Bob Hancock discovered a whole lot of these cationic peptides. Some of them can be taken orally, some of them are injected, and there's a lot of research going on with that right now. So he donated all of the naturally occurring antimicrobial peptides to the World Health Organization as a service to humanity, and then he kept a few of the artificial ones to himself to make money. That's, that's probably the ideal scientist. You don't want to be, just because you're a scientist doesn't mean that you have to starve to death and be poor all your life. And and most of them are not, or some of them are not anyway, unless, well, some of them are, but some of them are not. But anyway, that was Bob Hancock's contribution to humanity, is finding another class of compounds that is capable of killing bacteria, because we're running out of antibiotics that work. All right, folic acid synthesis. All bacteria need folic acid as part of their membranes. Humans get folic acid from their food. Bacteria generally make their own. If you can find chemicals that will inhibit the enzymes that make folic acid in bacteria, you will have found the ability to kill most bacteria. Generally, they tend to be a little bit bacteriostatic and not bactericidal. Okay, so the sulfonamides are inhibitors of folic acid synthesis, right? These are referred to as the sulfa drugs, right? So the sulfonamides, also known as sulfa drugs, are, like the name suggests, they contain sulfur. They are usually a yellow powder, the sulfa drugs. And they are bacteriostatic, meaning that they don't permanently kill these bacteria, but they stop them from growing, and they give your immune system a chance to catch up and kill some of them, and then maybe you can try something else, a different drug as well. But they're convenient and they're inexpensive. And so an example would be sulfadiazine, right? Sulfadiazine. All right, now, if you ever see old American war movies about World War II and stuff like that, you'll often see in these old American war movies, somebody will be out on the battlefield and they'll get wounded, they'll get shot through the leg or something like that, and then they'll yell medic, and the medics are these guys with the red cross on their helmet. And you'll see a medic will run out with a pouch with a pouch under his arm, and he'll pull out a little package and tear open the package and pour this yellow powder all over the wound. That yellow powder doesn't do anything to heal the wound or to stop the pain, but what it actually does is it stops the wound from being infected, because the yellow powder is sulfadiazine, or sulfonamide powder rather. Right, so it was a bacteriostatic compound. It prevented wounds from being infected for a short period of time, and then they would use a more thorough method to clean out the wound later on. Okay, now the concept that I've been talking about all along here, antibiotic synergy and antagonism, is kind of interesting. So some antibiotics together will work better than either one of them could alone, and it's usually because they're attacking bacteria using two different mechanisms. There's some bacteria that we say are neutral because they're both acting upon the same mechanism, and so having the two of them together doesn't work any better than if you simply add twice as much of either of them. Right, so that would be a neutral interaction. But there are some that actually work against each other because they're working on two different mechanisms that kind of are necessary for one of them to work. All right, so if you mix together different types of antibiotics, some of them work together in synergy, some of them are antagonistic. Right, so synergisms. Example, if you mix together the penicillins and the cephalosporins, or penicillins or the cephalosporins, and the aminoglycosides, right? One of them inhibits protein synthesis, the other one inhibits cell wall formation. They would probably be synergistic. Antagonisms. If you mix together penicillins and cephalosporins, for instance, with ciprofloxacin, the penicillins and cephalosporins can only kill the cells when they're dividing, and the ciprofloxacin is actually blocking the cell from dividing because it's blocking the replication of the DNA, right? So that if you add the ciprofloxacin, it would essentially nullify, it would neutralize the penicillin or the cephalosporin, right? So that's an example of antibiotic antagonism. And then neutral, if somebody takes penicillins together with cephalosporins, doesn't matter because they're both doing the same thing. And so if you took twice as much penicillin, it would be the same as if you took penicillin plus cephalosporin, right? So these are the three different relationships that are possible. Okay, now here's how you would test it. You remember this test. This is our friend the Kirby-Bauer test, right? So here we have two different antibiotics in two different filter paper discs. So here's antibiotic A over here. It has a clearance zone that's that big, right? So we can measure the clearance zone, and it's only that big for that antibiotic by itself. Over here we have another antibiotic that by itself the clearance zone is only that big, right? But notice that in between them there's an even bigger area, and that indicates this is an area of antibiotic synergism, right? So the two antibiotics are working better in this area because they're together than either one of them does alone, right? So that's a very simple, elegant way to test for antibiotic synergism. All right, here's another example. This here we have a really lousy antibiotic over here that by itself it does practically nothing. You can see there's practically no clearance zone. Over here we have a good antibiotic that has a fairly wide clearance zone. And in between them you see that this one actually picks up, it gets a clearance zone as a result of being combined with this other one, right? So these two are synergistic. Okay, this one here, notice that this one has a nice round clearance zone to the left, but the zone where the chemical is from this disc is actually pushing the clearance zone, kind of cramping it this way, because this antibiotic is actually interfering with the effectiveness of this one. So that's an elegant example of antibiotic antagonism. Right. Okay, so try this exercise. You tell me if you think each of these will be antagonistic or synergistic. Right, so penicillin G, penicillin V, would it be synergistic, antagonistic, or neutral? What do you think? Okay, the answer is that would be neutral because they're both the same thing. Right? How about penicillin and ciprofloxacin? All right, the answer is that would be antagonistic. That would be an antagonistic relationship because the ciprofloxacin is stopping the cells from dividing, and the penicillin can only kill them if they are dividing. So the ciprofloxacin is still working, but the ciprofloxacin has essentially knocked out the penicillin. Right, methicillin and polymyxin B. Right, I would guess polymyxin B punches holes, so it would probably. What do you think? That would probably be synergistic, I think, because they're working on two different mechanisms. The methicillin is preventing the cell wall from being formed, and the polymyxin polymyxin, as I said before, has a difficult time getting at the plasma membrane for gram-positive cells. Right? So polymyxin B tends to be more effective with gram-negative cells, and if you have, because the plasma membrane is more easily accessible than it is in gram-positive cells. So I would say those two are probably very synergistic. All right, what about penicillin V and ampicillin? Right, that would be neutral, right, because it's really the same thing. One of them is an artificial penicillin. Ampicillin is an artificial member of the penicillin family. Not something you would use in a pill because it's very toxic. All right, methicillin and sulfadiazine. Okay, I would think those two would be synergistic because methicillin is preventing the cell wall from forming, and the sulfadiazine is inhibiting the folic acid synthesis. That would either be synergistic or neutral. If it is synergistic, probably not that much because they're working on two completely different mechanisms, but by attacking one mechanism, you're not actually helping the other mechanism. Okay, so we talked about the penicillin family, the real natural and the synthetic penicillins. We talked about the completely synthetic penicillins, which are the cephalosporins. We talked about vancomycin and bacitracin, which are two other methods that prevent bacterial peptidoglycan cell wall synthesis. We talked about isoniazid and ethambutol, which inhibit mycolic acid synthesis. We talked about these other ones that inhibit the ribosomes, and we talked about polymyxin B and sulfadiazine drugs. All right, so that was all of them. All right, now how do we test the effectiveness of antibiotics and how do we test toxicity to humans? All right, so the effectiveness, we use two different methods, or three different methods actually. One of them is called the Kirby-Bauer test, which you already know about. You put the filter paper discs on a plate that has bacteria growing on it. The E-test strip is actually a little bit more specific. It's related to the Kirby-Bauer test, but a little bit more specific. Right? And then the broth dilution test is simply a serial dilution of the antibiotic inside a microtiter plate to see how much you have to dilute it before it doesn't work anymore. Right? Okay, so we're going to talk about three methods of testing the effectiveness of antibiotics, namely the Kirby-Bauer test, the E-test, and the broth dilution test. And then we're going to talk about two methods of measuring toxicity for antibiotics. One, you already know what the ED50 of an antibiotic is. And so you would do experiments to determine the ED50. I mean, in other words, if you take a bunch of guinea pigs, you would infect a bunch of guinea pigs. Well, you can do. I'm sorry that you wouldn't necessarily need to use guinea pigs here, but you can take just in a test tube. You can take antibiotics and you can mix them with bacteria and see what dosage do you need in order to kill half of the bacteria. You can also do the same thing with animal experiments, and then you determine the effective dose. This is a way of determining how much of that antibiotic do you need to use in order to kill half the bacteria, either in or out of an animal, like a human, for instance. And typically the ED50 is measured in milligrams per kilogram body weight, by the way, right? So if you have a 100 kilogram human and the effective dose is like 50 milligrams per kilogram, you would have to give that human 50 times 100 milligrams of the antibiotic. Right? So generally effective dose is measured as milligrams of the antibiotic per kilogram of body weight of the person or the animal that you're putting the antibiotic into. Okay, then you definitely need to use animals to determine what is the toxic dose. Right? So if you have a bunch of guinea pigs and you give them various amounts of the drug, then you figure out how much do you need to give them before you kill half of them. Right? So that is the TD50. Right? So the TD50 is also usually measured in milligrams per kilogram of body weight. Right? Now, this last number, you definitely need to know about this. It is called the therapeutic index. Right? The therapeutic index is the ratio of the toxic dose divided by the effective dose. Right? Now, it stands to reason that if the toxic dose is much greater than the effective dose, that antibiotic has a very low toxicity, doesn't it? Right? So if you only need to use, let's say for example, you only need to use 10 milligrams to cure a person of the bacteria, but you would need to give them 1,000,000 milligrams to kill them, right? So that means that the effective dose is much, much smaller than the toxic dose, right? So the amount of antibiotic that you need to give to the person in order to cure them is much, much, much smaller than the amount that you would need to give them to kill them. That drug has a very high therapeutic index, a very high TI. And those are the antibiotics that you want to look for. Right? So typically penicillin the Penicillin family has a therapeutic index which is in the thousands, 30,000. Right? So that means you would have to give somebody 30,000 times you would have to give somebody 30,000 times the normal dose of penicillin before you would kill the human, right? So the amount of penicillin that you have to give to a human to cure them of a bacterial infection, you would have to give them 30,000 times that much in order to kill them. That means that penicillin is a very safe antibiotic, and that's why we haven't completely abandoned, Penicillin family drugs, because they are very safe. They're very non-toxic. As opposed to things like vancomycin, which have a therapeutic index of, I don't know offhand what it is, it's around 10 or something like that. So if you gave somebody 10 times too much vancomycin, you could kill the person instead of just the methicillin that you're trying to get rid of. Right, so be aware of what the therapeutic index is. What is the ED50? What is the TD50? And what is the TD50 over the ED50? It's the therapeutic index. A drug or an antibiotic with a very high therapeutic index is very safe and non-toxic, whereas the lower the therapeutic index is, the more toxic the antibiotic is. All right, here's our friend the Kirby-Bauer test. So you put filter paper on discs and it diffuses out onto the plate. You have spread plate, Staphylococcus aureus or something all over the plate, and then you measure how big the zone of clearance is in order to quantify how effective the antibiotic is. This is the E-test, which sort of does the same thing. What you do in this case is you take a strip of filter paper and you impregnate it with different concentrations of the antibiotic, right? And then you see at which point does the bacteria hit this little ruler, right? So this paper is measuring the dose of antibiotic that's in the paper at various levels, right? So you can see that eventually you get to a point where the dose of antibiotic that's in this piece of paper, this stripped, or this E-test strip, is the bacteria is no longer killing the bacteria. Right, so that's telling you that a concentration of 64 milligrams is not enough to kill this particular bacteria anymore, whereas 250 milligrams is plenty to kill it. Right, so the Kirby-Bauer test quantifies, I keep using the word quantify. The word quantify, the difference between quantify and qualify is the difference between a quality and a quantity, right? So if you want to quantify the effectiveness of something, you need numbers, right? They want numbers. That's what quantify means, right? So how am I going to get numbers so that I can compare the effectiveness of penicillin G to the effectiveness of chloramphenicol, right? How am I going to get numbers? Right, so one way you can quantify the effectiveness of two different antibiotics is by doing the Kirby-Bauer test, and you measure the size of the clearance zone in millimeters, right? So that gives you numbers. And the E-test strip also gives you numbers, and it gives you exact number. That exact point where the bacteria touches the E-strip is the point where the antibiotic is no longer effective at killing the bacteria. So that's a very good number to have. That's a very good way to quantify this particular antibiotic. Okay, broth dilution. You take some LB broth and you put it into the wells of a microtiter plate. And then you take an antibiotic and you do a serial dilution of the antibiotic, and you put the different concentrations of antibiotic into those wells, and then you put bacteria into all those wells, and then you'll see that for certain concentrations the antibiotic will prevent the bacteria from growing, and then at some point it won't, right? And so then you'll see cloudiness, you will see turbidity, right? So you'll see bacterial turbidity in the well. So if you put that microtiter plate into the microtiter plate reader, you can very quickly, and you set it on 600 nanometers, you put it into the microtiter plate reader and set it for 600 nanometers wavelength of light. You can very quickly read the results of many different antibiotic tests using the plate reader. That's basically what a microplate reader does. It allows you to quantify how much stuff or how much color change there was in each of these 96 wells in a few seconds, basically, right? So that's called the broth dilution test. Right, so you can see here on the bottom, these are two different antibiotics, ethambutol and kanamycin, right? So you dilute. So at these concentrations, the ethambutol and the kanamycin can kill the bacteria, but once you get to this dilution, it no longer does, right? Whereas streptomycin is clearly more powerful with this particular antibiotic because it kills through all of these, and sulfamethoxazole is less effective still because there's some bacteria growing there, right? So if you get no bacteria growing until the fourth well, that's good. If you have no bacteria, that would be the case of kanamycin. If you have no bacteria growing except until you get to the fifth dilution or the sixth dilution, that's much better. All right, so the effective dose, how much do you need to add to kill half the bacteria? That's a number. That gives you a number in milligrams. The number is usually milligrams per kilogram body weight of whatever you injected the drug into. And then you get how much do you need to inject this animal with in order to kill half of them. This is a test you would do with guinea pigs. Sometimes you do, at some point you have to move beyond guinea pigs to humans. You usually do the testing with guinea pigs, and then at some point they do things called clinical trials. One of the first clinical trials that they do is they determine the lethal dose of a drug on humans, and generally it's sad to think about, but that usually is humans who are terminal, right? So people who are going to die anyway have volunteered to let them do this research so that they can save other people later, and the drug may kill you if you volunteer for that, right? So that's one of the, I believe, I don't remember whether that's phase 1, 2, or 3 clinical trials where they do that. All right, so examples. The therapeutic index for the penicillins and the cephalosporins is in the thousands, possibly up to 10 or 20 or 30,000. Antibiotics with low therapeutic indices, which are therefore more toxic, include vancomycin, polymyxin, chloramphenicol, the tetracyclines, and the aminoglycosides like kanamycin, for instance. All right, so remember that we're dealing with selective toxicity. We learned about several different key antibiotics. We learned about the Penicillins family, the Cephalosporin family, which are the artificial penicillins. The isocyanid drug is an example that will inhibit mycolic acid synthesis. The tetracycline family, we learned about rifampicin, polymyxin B, and the sulfonamide family. And so we learned about all of these different various antibiotics and how they work. Right, we learned how to measure the effectiveness of antibiotics and also how to measure and quantify the toxicity of antibiotics using the therapeutic index. Right? And we talked about the rise of antibiotic resistance and how to prevent it by not taking antibiotics, or how to slow it down at least by not taking antibiotics when we don't need to, by not abusing the use of antibiotics, and so on. We learned about the new areas of research, the cationic peptides and the antimicrobial peptides. All right, now as a bonus to this lecture, I'm going to give you just a minute introduction to bacteriophages. Right, so bacteriophages is a very new, very experimental method of killing bacteria. So we've got to kill bacteria in our toolbox. We now have antibiotics. We have cationic peptides, we have microbial peptides, and now we're just starting to develop something new, which are the bacteriophages. All right, so bacteriophages, this is a very new experimental technique. Bacteriophages are viruses that only infect bacteria, so therefore they have a very narrow host range. Right? They have a very narrow host range, they only infect bacteria. There are some, as you now know from last week's assignment, you know that there are lytic bacteriophages and there are lysogenic bacteriophages. Lytic bacteriophages do not cause, they infect bacteria, but they don't cause them to burst, but they do sometimes move genes around from one bacteria to another. And you now know that oftentimes virulence factors are moved from one bacteria to another using temperate bacteriophages. Right. Now, so bacteriophages can be temperate or lysogenic the way any other type of virus can be. So there are lysogenic bacteriophages that get into bacteria and then they just burst them because they produce so many progeny bacteriophages. But they don't affect the humans that these bacteria may be in. And so they have a very high therapeutic index. Right? So bacteriophages tend to have, at least the ones we've looked at so far, we haven't examined all of them yet, but the ones we've looked at so far have a very high therapeutic index. They are very non-toxic to humans, right? So at the moment, research into bacteriophages are commonly known as phages. The technical name is bacteriophage. what we're doing now is doing research into something called phage therapy, where if you get infected with a bacteria, they can give you phages, they can inject you with phages instead of antibiotics. This has been done on a kind of an emergency basis where they found people who are infected with bacteria that are resistant to every known antibiotic. It's going to kill you anyway, so what do you have to lose? Why not try bacteriophages? A few people have tried this and they've miraculously been cured of superbugs by bacteriophage therapy. But we don't know all of the bacteriophages yet, so the current areas of research are to hunt for previously unknown bacteriophages, and then use genetic engineering to modify the bacteriophages that we already know about, convert some of them from temperate phages to lysogenic phages. The bacteriophages tend to have a very narrow host range, so we believe that you can find bacteriophages that will only infect Streptococcus pyogenes, and some that will only infect Staphylococcus aureus, and some that will only infect E. coli, and so on. And so if you target somebody with a bacterial infection with a specific bacteriophage that will leave the good bacteria in their intestines alone while killing the Staphylococcus aureus that's infecting your blood, that's kind of what they're working on now. And in some places in the world, bacteriophages have already been licensed as a food preservative. I don't have a complete list of the places where they've done this. I believe California is one of the places. But if you go to one of these places and you buy meat, they, instead of filling the meat up with antibiotics to preserve it, or instead of pouring toxic powders onto the meat to preserve it, they have actually sprinkled it with a little bit of bacteriophage to kill any bacteria that may grow on it. Right, so that's bacteriophages. That is going to be the next wave of the human battle against bacteria. So here we have a diagram of what a bacteriophage looks like a virus. If this was a virus, which it is, we would say that it has a complex capsid. It's a naked capsid made of protein. It has a capsid head, which is made of an icosahedral capsid. There's a sheath and a shaft, and then a plate. These things will kind of land on the surface of a bacteria, and then through this sheath, they will inject their genome into the bacteria. The bacterial machinery is then taken over to create more of these bacteriophages, which bursts the bacteria. So over here on the right, we have a false color SEM. It's actually a false color. It's a shadowing technique. It's a false color electron microscope image showing thousands of these bacteriophages invading a bacterial cell and then bursting it. After it gets burst, it looks like this. So here we have, this is a false color SEM. So you can see that they have colored the bacteria green, and they've colored the bacteriophages purple. And you can see in the middle, this is what happens when this bacteria is infected with a lysogenic bacteriophage. Okay, so that's it. We've mentioned the antibiotics. We've talked a little bit about cationic peptides and antimicrobial peptides, and we've talked a very little bit about bacteriophages as the next wave of the battle of humans against bacteria. All right, that's it for the course. Thank you all for listening, and good luck on the final exam. Good luck to you all.